Now going to topic mitochondria. This one is the one of the cell organelle membrane. Mitochondria and chloroplast, plasma membranes, Golgi complexes, these are the cell organelle, different type of the cell organelles. So first of all, mitochondria, this is the just like it is a thread like structure. It is thread like structure. Mitochondria means thread like structure. Mitochondria. It in this type mitochondria name, it is in Benda. So in before of in this Kolliker he announced in 1880. It is a just first time it is the first time constructed, it is known as mitochondria. After Fleming, W. Fleming, he discussed that is the different type of discoveries in the mitochondria. First of all, he is only this C and discovered, but he is not given a sufficient name, not given a suitable name. Then after W. F. Fleming is also that it gives to the name mitochondria. So then Altman, after in both after then Altman and Benda, he is discovered, Altman and Benda. They discovered different type of thread like structures. It is called to the mitochondria. It is announced in 1890. It is announced in 1890. So here by different type of then the morphological studies, that is a different type of the morphological studies in almost all, it is a culture, that is then different type of the structures studied with the help of the different type of the microscope. It is studied about it is the underestimate or under observe the microscope, that is the electron microscope. The mitochondria is observed by the electron microscope. So it is start with then the help of the complete different things. It is a powerhouse of the energy. So it is in then the mitochondria, it is a different type of conditions, the different things, it's also different type of the things also. Then it is depending completely different certain type of the functions. It's change their completely shape, structure, structure is also most also different type of the things. So most of it is then the then different type of the membrane, that is going to the different it's depending certain helps of then many changes their shape, and it's take place completely different type of then the size. Then the maximum size of then the mitochondria, it gives to 0.0 micrometers, 0.0 micrometers to 2 micrometers. Then different type of the things, that is the under the microscopic section. So then from complete there may be in mitochondria of in each one of the cells almost all 500 to 5000. In sometimes some cells it is in 50,000 cells also. 50,000 cells also it has. 500 to 5,000 minimum cells, minimum mitochondrial cells in each plant. Then some of in higher plants they have to the 50,000 cells also they have. Then is the complete, then they show by the quite opposite of then the shows then the smooth of outer, then the different type of then layer, thick layer, at the same time it has into the different type of the layer, that is the thick layer and thin layer, that is the outermost layer, at the same time innermost layer. It has in inside and the separate from the membrane, that is the mitochondria. It is an observation by the electron microscope. It is a thick and thin layers respectively, inner layer and outer layer, inner layer and outer layer. So this type of the cells, it is going to the different type of parts, the different type of parts, almost all it has in 2 layers. Mitochondria, it has in 2 layers. For example, then two layers of the mitochondria. It has different type of the parts. Then different type of the matrix type of the layer. So it is called to the matrix. This one is called to the matrices. So it has most of the different type of then the particles, that is then the different type of the particle. Then again it one is the outer layer or outer membrane or outer layer. Then this one is the inner layer or inner membrane, inner layer or inner membrane. Also it has most of the different type of the things, that different type of things, that means ribosomes as well as DNA. The most of the ribosomes and DNAs, different structures of the ribosomes. Ribosomes. Then different type of structures, that is the DNA structures. Then complexes, different type of the smooth or rough type of the cells. General things of the mitochondria. So it has most of that different type of then structures. It has in oxysomes, it called to the different type of the oxysomes. For example, it formation to the different oxysomes formation. Then the oxysomes formed different. So that different, this means outer, this one means inner layer. So here it is the oxysomes formation, that breakdown of then the oxysomes here, and the formation like this. Respiration of oxysomes. Then here it is releasing to the formation oxysomes. So here it is resolution. Another, then it is in the resolution. The formation to the resolution, it is going to the resolutions. Then these are the completely formation of the oxysome formation. Then takes place of the then the mitochondrial crystals. Then it is in the different type of the structure of then it is in the small post of then the RNAs and DNAs. So here it is a DNA and RNA, then different complete then the different type. So here the RNA it is the different, that is the tRNA, at the same time polymerase RNA. tRNA, it is in the tRNA. That is the polymerase ribosomes of type of the polymerase or ribosome RNA. Then it has then the complete then the type of the circular type of the liner shapes. Circular type of liner shape. Then the gene, it is contained completely then the mitochondrial genome, that is the empty DNA or mitochondrial DNA. It is called the gene. Whenever the gene it is composed to the mitochondrial structure, mitochondrial genome, it is called to the empty DNA or mitochondrial DNA. So here it is the completely proteins or complete synthesized. Then proteins are synthesized. Proteins are synthesized. Then it is in the different thing, that different type of then the different type of inner mitochondrial membrane or at the same time outer mitochondrial membranes. Then it functioning or it is in gradual type of then the different type of the things. Then the osmosomes formation, then the osmosomes whenever they can the resolution and the different type of the things of the formation is different of the thing. So here then the breakdown of then this one. So here it is the separation of the cell. Then it is going to the different thing, then uncover each of the cells. So these are the different. So whenever the resolution is completely combined with the together, then the structure is formation after as usual structure of then the structures. Then this mitochondria, it is completely it is a protein mitochondrial. It is first bind of the, that is the different type of the proteins. It is the mitochondria, it is in the different structure of the protein, different structure. Then it's depend in the layer of then the epidermis layer, it is called to then the cytoplasmic type of the layer. Then it has then the epidermis layer, at the same time the cytoplasmic layer, it is the head type of the mitochondria type. Then the sides of then the face of M phase, or it is in the face of then M phase. Then the cytoplasmic Then cytoplasmic or cytoplasm structures, then here it is gives to the diplomatic and then covered by this, then almost all of the 60 angstroms to 80 angstroms. Then it has in then the thickness of then the matrix at the as usual of the its participation of then the electronic microscope particles we can observe and choose then the oxysomes also formation. Then it is in the fusion of then it is the formation of the nature type of the mitochondria. Then the nature type of the mitochondria, it has in completely then empty DNA production, its own mRNAs, tRNA or rRNA. It is production to the empty DNA production. At the same time, its own mRNA, its messenger RNA. At the same time, transfer RNA, tRNA, mRNA and tRNA at the same time rRNA. Then different type of the RNAs is also it's produced. Then the mitochondria process completely, then 75 angstroms of ribosomes is present. 70 the ribosomes, 70 angstroms of the ribosomes, that is then the formation, it is dividing into the 50 plus 30. Then the formation of different type of the structure proteins, structure of the proteins. Then the Armstrong of then the mitochondrial, then presence of then enzymes. With enzymes is completely required to the functioning. It's complete then the male or any kind of the sterility of then different type of then the it's then the mitochondria, it is also developed different type of the fusion of free structure of the or free mitochondrial structure, that is the particle completely then the controlled. Then the young stems or it is present in the stems or leaves or leaf sheaths. So then the fully autonomous of both the structure, it's partially complete then the available material from symbiosis of the eukaryotic cells, as well as then become associated with the evolution in different type of the cells. So here then the different type of then the functions, that is the different type of function, it is mostly in the respiratory functions. It takes place by the mitochondria. It is in electron transport system. At the same time, it has in crystals. So it is in the most of the used in the functions of the mitochondria. Then the functions of mitochondria. First of all, it most of the used in respiratory system. Then again it used to the Krebs cycle, activation of the Krebs cycle in matrix, activation of Krebs cycle, activation of the process. Then it is known as already first I mentioned that it is known as powerhouse. It is known as powerhouse. So it is most of the thing it is mitochondria complete exhibit discovered the Kolliker Altman and different type of the Benda Altman, and all together they are discovered by the mitochondria in different type of the consist of then the membrane, it is called to the perimitochondrial. It is also then the peripheral layer consists of the peripheral layer, so it is also known as to the perimitochondrial membrane. So it has in the two layers of formation, that is a thick layer and thin layer, that means the outermost layer and at the same time innermost layer, and this one is the formation of the different type of the formation of isomerases. So then the oxygen, that means then the isomerases, then breakdown of then the stage, then this one is the isomerase stage. So here then the mitochondria, most of it is in the protein synthesis, as well as it produces to the DNAs. At the same time it is it has to the different type of the RNA structures. That different type of RNA structures is given to the mitochondrial, that means messenger RNA, transfer RNA, respiration, or different type of then the mitochondrial DNA is also produced. Then the mitochondrial DNA, almost all it is called to the meta DNA or mitotic DNA. Then its presence in most of all the function, the main important function that things is the respiratory systems. So it is in the most of used to the in respiration system. That then again it is used to the different type of the activations of used in the Krebs cycle. Then it has in used in Krebs cycle also. Then it is also produced to the ATP is also produced by the chloroplast synthesis. ATP also produced in synthesis. So 5% of then the during of then the that means anaerobic respiration, it is in anaerobic respiration, during to the anaerobic respiration. So most of the this one is the in mitochondria, study about the mitochondria structure and isomerase formation mechanism, the general introduction of the mitochondria, structure of mitochondria, formation of isomers, and functions of the mitochondria. So this is the today topic.